Dr. Stephen Meyer is a geophysicist and Cambridge University PhD in philosophy of science. He is a New York Times best-selling author and director of the Center for Science and Culture.
Do we have in the simple cell an appearance of design? In fact, an appearance of design that has not been explained away by purely unguided, undirected materialistic processes. We do. And the most striking appearance of design, or at least one of the most striking appearances, there are many at the microscopic level, is the presence of this digital code, especially when we understand what it's doing. Richard Dawkins says that the machine code of the genes is uncannily computer-like. Apart from differences in jargon, the pages of a molecular biology journal might be interchanged with those of a computer engineering journal. We have digital code, we have replication, all this intricate information processing system. Bill Gates, our local hero out in the Seattle area, says DNA is like a computer program, but far more advanced than any that's ever been created. Well, at the very least, from these statements, we can see that we have a striking appearance of design. The question is, has that appearance, the appearance conveyed by the digital information in DNA, been explained by any undirected, unguided, materialistic process? Such that we could say, as Darwin said of at least minor adaptations in life, the appearance of design has been explained away by a mindless, materialistic mechanism. Now, in signature in the cell, and I'm not going to go through all of this in this talk because I don't have time. I'd love to, actually, but it's also in the book. I look at basically there have been three different approaches to trying to explain the origin of the information necessary to build the first living cell in an unguided, undirected way, within a materialistic framework for doing science. There's a famous book by Jacques Monod, who was one of the great molecular biologists of the 1960s, and it was called Chance and Necessity, and it was basically a manifesto of the scientific approach to things. And he was saying, look, if you're going to be a scientist, you've got two tools. You either got to explain things by chance, by random, undirected, what are sometimes called stochastic processes, or you can explain things by necessity, where necessity relates closely to the concept of natural law. If I drop a ball and it falls to the earth, we say it falls because of the law of gravity, or we could say it falls by necessity according to the laws of gravity. And Monod is saying, you want to explain the origin of life, you got to explain, or anything in science, you got to do it by reference to chance, necessity, or, as in Darwinian theory, the combination of the two. The law-like process of natural selection acting on random, undirected variations. And I'm going to just briefly touch on some of the problems with each, and then I want to turn the corner and look at another approach to explanation, another possible explanation. Okay, you heard last hour about Doug's work on proteins and how incredibly improbable the functional proteins are in relation to all the possible ways of arranging amino acids. Okay, how many of you, most of us are familiar with ordinary bike locks, right? So if you got a bike lock with 4 dials, how many possible combinations do you have associated with those 4 dials? A lot of people think, if they're not thinking, they'll say, well, 10 plus 10 plus 10 plus 10, you got 40 possibilities, 4 dials, 10 at each. No, it doesn't work that way, does it? Because if you just look at the first 2 dials, you got 10 times, each one of the dial settings in dial number 1 can match up with 10 others, but there are 10 such settings that can match up with 10 others. So you got 10 times 10 possibilities for 2 dials. 3 dials, it goes to 10 times 10 times 10, or 1,000. 4 dials, it's 10 to the 4th, or 10,000. So you have this strange, this is a problem known as combinatorial inflation. As you increase the number of dial settings on the bike lock, you increase the number of possible arrangements exponentially. So if a thief sees a lock over here, or a bike all locked up, and he sees one with 3 dials and one with 5 dials, which one is he going to choose, even if he's not taken any math courses? He knows intuitively. He's got a chance, but 3 dials, that's only 1,000 combinations. I mean, if he keeps at it, the police don't come around the corner too quickly, he might get that one by chance. But the 10,000, now what if you had a bike lock like that? Okay. My artist in the a new book coming out, has drawn a bike clock with 100 dials. And now we're getting close to doing what Doug does. Okay, now we're talking proteins. Doug has worked with a system, a protein with 150 amino acids, a protein domain, just a part of a protein that makes a fold, 150 amino acids long. And on our protein bike lock, we don't have 10 possibilities at each, we've got 20. So what kind of numbers do you get? What kind of, as far as number of possible combinations? Well, if you've got a protein, a little short peptide chain that only has 10 sites, then you got 20 amino acid possibilities, you've got 20 to the 10th possibilities. But what if you deal with what Doug's dealing with, and you are looking at a 150 amino acid protein domain? You've got 150 sites with 20 possibilities, or 20 to the 150th power, which computes in base 10 to 1 chance in 10 to the 195th power. Or rather, let's put it this way, forget the 1 chance in bit. It's 10 to the 195th combinations. Now, to know how many of those, how probable or improbable it is that you'd stumble on one of those, you have to know what Doug knows. You have to do the experimental work that Doug did to find out how many of, in effect, we could think of back to our bike lock. You could imagine a bike lock where there was more than one combination that would open the lock, okay? But also there were so many combinations that even though there was more than one combination, your odds of cracking the combination would still be minuscule. And that's essentially what's going on in proteins. It's what Doug has shown, is that even though there is more than one functional combination of amino acids corresponding to that big space of 10 to the 195th possibilities, the ratio of the ones that will open the lock to the ones that don't open the lock is incredibly small. You can't, it's mind-bogglingly small. You can't get your mind around it. It's that 10 to the 77th number. Now, what I did in signature in the cell was I took Doug's number for getting even a single protein, 1 in 10 to the 77th power, and then I multiplied it by other things that had to happen that were also incredibly improbable, like getting amino acids that had the correct orientation called chirality, and getting the right kind of connections between the amino acids, getting the right kind of bonds. And I got a huge scary number that the chance of building a single protein by chance since the beginning of the universe was 1 chance in 10 to the 164th power. But then you had problems like there's only 10 to the 80th elementary particles in the universe. And so without going into all the numbers, and I can direct you to the book where I do all that, here's what I came to. And who asked the Jim Carrey question in the last hour? That was brilliant, okay? Okay, because here's the way to think of this. What I showed in the book was that if every event, where an event is defined as an interaction between elementary particles, if every event since the Big Bang, assuming 14 billion years of the standard calculation of the age of the origin of the universe, if every event since the origin of the universe had been devoted for looking for one of those amino acid combinations in that big space of possibilities, you would only get to search 1 10 trillion trillionth. It was an incredibly small fraction of that space, okay? So when you're trying to figure out a probability, whether a probability is plausible or not, and this gets to the Jim Carrey question, it's not a question of what's the first number you get. Okay, it's really improbable. It's really improbable that on the first try, I'd open that bike lock. Now, that's not enough. You need to know more than that. You need to know how many opportunities you have to open the lock, right? How soon is the cop coming around the corner? Okay, that's the calculation that the thief has got to make. Okay, so let's say we're back to one of my bike locks, okay? Let's say he's dealing with that one, all right? And he's got 3 minutes before the security guard comes around the corner. Here's the question. Is it more likely than not or less likely than not that given that amount of time and the probability he's got a crack, that he's going to open the lock? And it's obviously vastly less likely than not, right? Okay? Now this is kind of a second order probabilistic reasoning. It's not only the probability of the event taking place, it's the probability of the chance hypothesis being true. The chance hypothesis is that he could open the lock in the time available. But since it's vastly more likely than not that he won't open the lock that way, it's also vastly more likely to be false. The chance hypothesis is vastly more likely to be false than true. And that's the answer to the Jim Carrey question. if a hypothesis is overwhelmingly more likely to be false than true, is it reasonable to believe it? Or more reasonable to disbelieve it? Do you see? Okay? So you can't, with the Jim Carrey, there's a chance. Maybe maybe you go out with, well, yeah, but it's overwhelmingly more likely that that won't happen. So if you're betting on what's more likely to be true or false, you bet against the chance hypothesis, okay? It's more reasonable to reject it. And by the way, folks, that is what every person working in origin of life research and biology has done. Nobody is any longer entertaining the chance hypothesis because the probabilities and the available, what are called probabilistic resources, the number of trials, are not sufficient to crack the problem, to open the lock, to build even a single protein in the known time of the universe, okay? So chance has really gone by the boards. There's another approach to this that has been proposed, which was really more Darwinian in scope or character, because Darwin never relied solely on chance. His mechanism was chance and natural selection acting together. But when you're talking about the origin of life, that definitely doesn't work for a very simple reason. Natural selection is a process that only kicks in. It only ensues once you have living organisms that are capable of reproducing. Natural selection is all about differential reproduction, okay? And this is essentially what's going on with prebiotic natural selection. In fact, Christian Deduve, one of the leading origin of life researchers and really great thinker on this question, Nobel Prize winner, says theories of prebiotic natural selection fail because they need information, which implies they have to presuppose what is to be explained in the first place. Now, there's one other approach to this, general broad strokes approach called self-organization. And that's the idea that you have some kind of physical or chemical law or force of attraction is responsible for the information in the DNA. So if you look at that formula for the DNA molecule I showed again, you think, well, maybe there's something that's making the A's, the C's, the T's, the different nucleotide bases line up the way they do. Maybe there's a chemical attraction that causes them to arrange into the sequences that they have adopted. And we know that there are forces like this in chemistry. There are molecules, the structure of which you can explain by reference to self-organizing forces of attraction. One really simple example is your table salt. It has the formula NaCl, Na plus Cl minus, attraction. You get a nice crystalline lattice, very orderly structure that is the result of self-organizing forces. But notice that's not what's going on in DNA. Look at the formula there. You can see the sugar phosphate backbones running up and down on the right, on the left. The nucleotide bases in the middle, with the representations as chemists represent them with the letters A, C, T, and G. And notice that between the bases, there are, also in these structural formulae, they have the sticks represent bonds. They represent chemical forces of attraction. Notice that between the pentagons and the phosphates, the sugars and the phosphates in the backbone, there are chemical bonds, okay? And notice that there are bonds between the bases and that backbone. But notice in the information-bearing axis, the vertical axis, there are no bonds between the individual bases. That is to say, self-organizational forces are not at work. There are no bonds, therefore the bonds can't explain the informational sequencing. Now that's possibly some tough sledding for chemistry, for non-chemistry people. So I've got an illustration. I've got a information-rich sequence here, although a little misspelling, I guess. Philly rocks, okay, I'm pandering to the local audience, all right? And there are forces of attraction at work here in this message system. There are little magnets in these letters, and this is a metallic chalkboard. And so the force of attraction explains why the letters stick to the backboard, okay? Why the message sticks to the medium. And in the DNA molecule, you have the same thing. You see you have the little sticks between the amino acids, or between the nucleotide bases and the sugar phosphate backbone. So there's a force of attraction that explains how the information-bearing characters are fastened to the medium in both cases. But does the information in this message, is that explained by the magnetism? What is responsible for the sequence? Notice no magnetism between the letters, just is there no chemical attraction between the characters. What explains the information in this message? at least a quasi-intelligent agent, okay? All right? So, and look, I can show you that the physical forces of attraction are not forcing this message to be constructed, right? Because look what I can do. Now I got gibberish, but I got the same magnetic forces in play. What changed? The arrangement that I imposed on the system, okay? And so this was one of the huge, the scientists who first proposed this self-organizational model eventually recognized that it did not work for explaining the origin of information in DNA. And so this model also has gone by the boards. What did create the information there? On my magnetic chalkboard, you obviously said it was an intelligent agent, right? Could the same thing be true of the information in life? And that's really where my story intersects this lecture. 1986, after having met Dr. Thaxton, I went off to grad school. I knew I wanted to work on this origin of life question. Thaxton had proposed in the epilogue of his book with his co-authors, Walter Bradley and Roger Olson, that perhaps what we were looking at in the living cell with the information in DNA was the product of he called it an intelligent cause. He said, because intuitively, information is it's a mind-like product. It's something that comes from that's what we know, he said, from experience. And so I began to wonder if this intuition, this intuitive connection between information and intelligence could be formulated as a rigorous scientific argument. And so that was my big animating question as I left for grad school. And I began to study the scientific methods and methods of reasoning that scientists use when they're investigating these questions about what happened in the remote past, in the distant past, these origins questions. And so naturally that led me to Darwin. And I learned that Darwin had a particular method of reasoning that is now called the method of multiple competing hypotheses, or the method of inference to the best explanation. And he said, in fact, in justifying his own theory, that he inferred his picture of the history of life as the best explanation, and he would hold it until a better explanation came along. But that raised a question, which was, well, what makes an explanation best? What makes an explanation best? And I came across an answer to that not only in Darwin's work, where he had a very specific criterion of best explanation, but also in the work of his scientific mentor, Charles Lyell, the great geologist. And I'll never forget the day that I was reading this boring Victorian subtitle to a dusty old book, and for me, the light bulb just went on. Here's the book, Principles of Geology. Now here's the boring subtitle. Being an attempt to explain the former changes of the Earth's surface by reference to causes now in operation. And I have to tell you, this hit me like a thunderbolt. I remember right where I was. Because what Lyell was saying is that if you're going to explain an event in the remote past, you shouldn't invoke a cause, the effects of which we do not know. You should invoke a cause, the effects of which we do know. You should invoke a cause that is presently acting or now operating, which has the power to produce the effect in question. The best explanation is by reference to a cause that is known to produce what you're trying to explain. Simple example, Eastern Washington, where I went to college. There is now, as of May 18th, 1980, a layer of ash out in the Palouse. Other stuff stacked on top of it, but you don't have to go very far down to find that layer of ash. What's the best, if you didn't know about the eruption of Mount St. Helens, and you're just a geologist working through those layers, and you come across a layer of volcanic ash, what is the best explanation for what caused that? Possible hypotheses. Might have been a flood, might have been an earthquake, might have been a volcanic eruption. Which one is best? Well, obviously the volcanic eruption, but why? Because we have experience of volcanoes producing that effect, and we have no comparable experience of floods or earthquakes doing the same. So as you weigh those competing hypotheses, you elect the one that has the known causal powers, to use some jargon from the field, to produce the effect in question. And so when I began to think about the information question in light of the Darwinian methodology, the Lyellian principles of uniformitarian reasoning, I realized that using Darwin's own method of reasoning, we should come to a very different conclusion about design. Why? Well, because there's something else we know about information. And there was an information scientist, and this was, in a sense, my second epiphany studying this. Information scientist Henry Quastler, early scientist, a pioneer in applying information theory and theoretical analysis to the molecular biology, to the genome. And offhand, not meaning to say anything about this big question of design in biology, he said the creation of information is habitually associated with conscious activity. Now think about that in light of causes now in operation. In other words, what he's saying is the cause now in operation for the production of information is what? Intelligence, mind, conscious activity. And so I realized that by applying Darwin's key standard of best explanation, Lyell's principle of reasoning, to the information question, to the DNA enigma, there was a powerful rationale supporting the inference to intelligent design. Why? Because we know of only one cause that produces information. I knew from my study of origin of life research that chance, necessity, the combination of the two, and all the models that fell under those mutually exhaustive categories had failed. But there was a cause, there is a cause of which we know that is capable of producing information. And that cause is conscious activity, rational deliberation, intelligent design. And is this consistent with our experience? Absolutely. If we go back to that computer code that Bill Gates was talking about, where does the information come from? It comes from intelligent agents. Now, I know some people have issues with Word, but you know, it's pretty impressive. We're in Redmond. Those are pretty smart people at Microsoft. You look at a newspaper headline, or a hieroglyphic inscription, or a radio signal embedding information in it, and you trace that information back to its source, you invariably come to intelligence, to an intelligent cause, not a mindless, undirected process. So when we encounter information at the foundation of life in these information-bearing molecules such as DNA and RNA and proteins, the most logical thing to conclude, based on our knowledge of cause and effect, based on our knowledge of causes now in operation, is that intelligence also played a role in the origin of the first life, because life depends on information. That's the argument of signature in the cell. I develop it in more detail, looking at each of these different nodes on the explanatory possibilities. Chance, necessity, combination of the two, but I argue that intelligent design based on our knowledge of cause and effect is the best explanation, the most causally adequate explanation. And I will take some the next hour is, in a sense, after the break, a continuation of this. So we won't do any Q&A now. I just want to close with a little story. There's a computer programmer that's worked very closely with Doug, and a brilliant guy, and he wrote the code that Doug was talking about in this stylus program. And he was a retired Microsoft architect, one of their elite programmers. Retired means he was about 40, okay? And he walked into my office one day, and he threw a book down on the table, and the book was called Design Patterns. He's 6'5", and he's got, you know, wild shock of hair. He's really brilliant, and he says, very dramatically, I get an eerie feeling that someone figured this out before us. And I said, what do you mean? He said, well, you need to know what a design pattern is. In my field, a design pattern is a proven strategy for solving a functional problem, solving a problem. And he said, these computer design problems are all about solving problems of information storage and processing. And he said, I'm having to learn molecular biology to write the code for your guys. And what I'm discovering is that in that information processing system at work in the cell, there are already design patterns that we've just been developing in the last 25 years in the software industry. And he said, that gives me an eerie feeling. But he said, I'll tell you what's even more eerie, is he said that all of the design patterns, they're the same strategies, the same recognizable engineering strategies, except they are executed with and he started talking Microsoft talk. He said with an 8.0, 9.0, 10.0 elegance that far exceeds anything that we have achieved. And then he started ticking these things off. he said, there's not only digital code. He said, I know that's what you're all excited about in your book, but he says there's also the nested coding of information, codes within codes. He said there's also a files within folders, hierarchical filing system. That's what we use on the desktop. He says we have that for storing the information in the genome. Then we've got the junk DNA is functioning as an operating system. And then we've got distributed storage and retrieval of informational modules, where on a needs basis, the cell goes out and accesses modules of information, concatenates it, puts together a new gene to produce a new protein for a specific application, so as not to burden the cell with an excessive amount of a storage problem. He says, we do the same thing. And on he went. So I gave this talk one time at a fairly hostile university, in a hostile university setting. And afterwards, there was a gaggle of students coming up, peppering me with questions. And there was a software engineer standing by, and he said, I was agnostic until you said the stuff about the design patterns. And then he said, I'm totally with you, So anyway, I leave you with that. It's not just, in a sense, the first 55 minutes of the talk were molecular biology 101. And in the book, I have an epilogue talking about what's being discovered about the informational structuring and patterning of the genome, and the design patterns that work in the whole information processing system. It's awesome, and I think it makes a whole series of additional compelling arguments for intelligent design, because each of these design patterns are known from one and only one known. There's only one known cause for these things. There's only one cause in operation that produces them, and again, it's intelligent design. Thank you very much.
To view this program again and share it with your friends, and to learn more about this series, these conferences, and the intersection of science and faith, please go to www.pensmore.org.
The preceding was brought to you by the Pensmore Foundation and NRB TV.